Today, I will explain how glucose enters the cell and goes through various metabolic pathways to produce energy and essentially intermediates. I will focus on glycolysis, glycogenesis, and the pentose phosphate pathway and the TCA cycle and show how they are all interconnected. There are also gluconeogenesis and glyconeogenesis pathway, but I will focus on glycolysis and glycogenesis pathway. I will discuss the key enzymes involved and whether ATP is used or produced in each step. The first, glycolysis. So glycolysis is this green-shaped one, and the glycolysis is the first step in glucose metabolism, where glucose is broken down into pyruvate in the cytoplasm. It consists of 10 steps during which 2 ATP molecules are consumed and 4 more ATP molecules are produced, giving a net gain of 2 ATP. So here are some important steps in glycolysis. So the first step is the hexokinase or glucokinase converts glucose into glucose 6-phosphate by using one ATP molecule. It is the first step. And in the third step, phosphofructokinase 1, in short PFK1, acts on the fructose 6-phosphate, converting it into fructose 1,6-bisphosphate. This step also requires one ATP molecule. So in the energy generating phase, the seventh step, the enzyme phosphoglycerate kinase produces one ATP molecule by converting 1,3-bisphosphoglycerate to 3-phosphoglycerate. So in the 10th step, and also pyruvate kinase converts phosphoenol pyruvate into pyruvate by generating another ATP molecule. So as a result, glycolysis produces a net gain of 2 ATP molecules and 2 NADH molecule, which is same with 5 ATP, and which will later contribute to ATP production in the electron transport chain. And then pyruvate can then enter the TCA cycle, as you can see here in the purple circle, for further energy production. Before I explain you about TCA cycle, or what is the Krebs cycle, I will explain glycogenesis first. So while glycolysis generates energy, glycogenesis is the pathway that stores energy by converting these letters glucose 6-phosphate into glycogen, which is stored in the liver and muscles. So when you intake feed, when animals intake feed or when humans intake food, and they have sufficient glucose, they don't have to use that, and they have to lower the blood glucose levels. So this glycogenesis pathway are activated, and this glucose 6-phosphate converts into glycogen by using this pathway. And they are normally stored in the liver and muscles as a glycogen form. So glycogenesis are as follows. So in the first step, the enzyme phosphoglucomutase converts glucose 6-phosphate into glucose 1-phosphate. Next, UDP glucose pyrophosphorylase activates glucose 1-phosphate by attaching UDP to it, forming UDP glucose. And finally, glycogen synthase uses UDP glucose to add glucose units to the growing glycogen chain. And this process allows glucose to be stored as glycogen. And when energy is needed, glycogen can be broken down back into glucose 6-phosphate. So this can go back into glycolysis steps, and then and then produce more energy for further like body functions or actions. So this shows that by directional nature of glycogenesis, where it balances between energy storage and release. And in these steps, the well-known hormone insulin and glucagons work as like regulating this process. So when there are too much glucose in the blood, insulins are released to decrease the blood glucose level by making glycogen. And when animals doesn't eat any food, so they have low blood glucose level, glucagon released, and they break down this glycogen to produce more glucose 6-phosphate and make it and use it as an energy source. The next step is this blue circle, which is pentose phosphate pathway. So the pentose phosphate pathway runs parallel to glycolysis and primarily generates NADPH and ribose 5-phosphate, which are important for anabolic processes like fatty acid synthesis and nucleic acid synthesis, respectively. So the key steps are the enzyme glucose 6-phosphate dehydrogenase converts glucose 6-phosphate in this red letter into 6-phosphoglucono-gamma-racton, producing 1 NADPH molecules, and these NADPH molecules equals to 2.5 ATP. The subsequent steps convert 6-phosphoglucono-gamma-racton into ribose 5-phosphate, which is necessary for the synthesis of nucleotides. And then NADPH produced in this pathway is crucial for protecting cells from oxidative stress and for biosynthetic reaction, because NADPH is work as reductant that takes electrons from free radicals. So when they take electrons from free radicals, they may decrease the chance that free radicals can attack the normal cells. And at the same time, ribose 5-phosphate is used for DNA and RNA synthesis. Because as you know, DNA is deoxyribonucleotide and RNA is ribonucleic acid. Thus, the pentose phosphate pathway can work simultaneously with glycolysis, redirecting glucose 6-phosphate based on the cell's need for energy or building blocks. So this blue circle pentose phosphate pathway is anabolic reaction, and also and then this glycolysis is catabolic reaction, but glycogenesis from pyruvate to glucose is anabolic reactions, which building up more complex molecules. But catabolic reaction is breaking down glucose into carbon number 3 pyruvate, so they are breaking down 6-carbon glucose into 3 carbons pyruvate to make more energy. And the last one is TCA cycle. I don't think the most, but important steps for producing energy, because you cannot use pyruvate as an energy source, but after pyruvate go through that TCA cycle, that can produce many ATP or NADPH for animal for their body functions. So after glycolysis, the pyruvate generated and then transported into the mitochondria, where it is converted into acetyl-CoA by the enzyme pyruvate dehydrogenase. And this is the entry point for the TCA cycle. And this TCA cycle is also known as the citric acid cycle or Krebs cycle, which occurs in the mitochondria and produces large amount of NADH, FADH2, and a small amount of ATP. And as I mentioned before, NADH and FADH2 is kind of reductant, so they can decrease the oxidative stress of free radicals in the body. And also they can produce more energy. So NADH equals to 2.5 ATP and FADH2 equals to 1.5 ATP. So this value has been changed a lot, but I think NADH equals 2.5 and FADH is equal to 1.5. The key steps in the TCA cycle include pyruvate is converted to acetyl-CoA by pyruvate dehydrogenase, producing 1 NADH in the process. And then acetyl-CoA then combines with oxaloacetate to form citrate, a reaction catalyzed by citrate synthase. And then the enzyme isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase generate 2 more NADH molecules in subsequent steps from citrate to isocitrate and alpha-ketoglutarate. And in another step, succinyl-CoA synthesis produces 1 GTP equals to 1 ATP, and succinate dehydrogenase generate 1 FADH2. As I told before, the NADH and FADH2 produced in the TCA cycle are used in the electron transport chain to generate large amount of ATP. Acetyl-CoA can also be diverted to fatty acid synthesis if needed, showing that the TCA cycle is not only for energy production, but also connects to another anabolic pathway, because acetyl-CoA have 3 carbons, but as you know, fatty acids have much longer chain than acetyl-CoA. So by using acetyl-CoA, they can make longer block, which is anabolic reactions. So as you can see, from glucose to glucose 6-phosphate, and this glucose 6-phosphate converted into glycogen when they are too much in the blood, and when they need to make more DNA, RNA, or other ribose things, this glucose 6-phosphate need to go through that pentose phosphate pathway. And for energy production, when animals are lactating or gaining their weight or making protein, they need to no, not making protein. When they need actions, they need ATP for use. So the glucose should be converted into pyruvate, and this pyruvate should go through this TCA cycle to produce more NADH, FADH2, and ATP. And if there are too much glucose that is used for energy production, this acetyl-CoA will go to fatty acid synthesis, and then you gain fat. That is how it works. So I skip this anabolic reaction, which is gluconeogenesis and catabolic reaction, glycogenolysis, but they are sharing same pathway and kind of similar enzyme, so I skipped it. Yeah. Okay, that's it. Thank you.